Assalamualaikum. Good morning, everyone. Welcome to Breakfast at FOM. I'm Sharifah. I will be your moderator for today, and your breakfast talk today will be served by Department of Pharmacology by our newest member of the edition, Dr. Hassan. So, Dr. Hassan, a brief introduction to Dr. Hassan. So, Dr. Hassan, before started his journey in Faculty of Medicine, he is a practice community pharmacy in Syria, while obtaining his master degree in biomedical engineering at University of Aleppo. He obtained his PhD from Faculty of Medicine, and he pursued with his postdoctoral degree fellowship. His area of expertise are in drug delivery, and his research interest is in nanomedicine, in cancer treatment, particularly polymetric nanoparticles, formulation, and the industrial scale of nanotechnology-based medicine. Without further ado, I would like to call upon Dr. Hassan to share his research findings.
Thank you very much, Dr. Sharifah, for the introduction. I actually tend not to prefer sharing my research findings in these brief talks, as more I would like to propose a framework or some theory that I learned from my research. So to share my screen. So what I would like to talk to you about today is cancer nanomedicine after the pandemic. So I worked, I had my PhD, started before the pandemic. I was about to finish through the pandemic, but it took longer than expected. One of the reasons is because of the pandemic itself. And I kind of went through the journey of what happened through the pandemic to nanomedicine as I'm doing my PhD. So first, I would like to give an introduction about the promises of nanomedicine, the sci-fi ideas about sending a miniature nano-sized robots that kill a specific pathogen or fix an ill cell specifically, or up to the level of sending miniaturizing humans and sending them into the body to fix problems. However, realistically, in the 2000s, things came up to this kind of approach to nanomedicine. So this is a real nanoparticle that was engineered by basically by these two gentlemen who will continue with us through this talk. The idea was to put your medicine inside a polymer-based nanoparticle. And ideally, this nanoparticle is non-immunogenic, so coated in a way that makes her stealth, hidden from the immune system. And on top of that hiding layer from the immune system, it has this small blue targeting molecules where they will make it home to its target. Go to specifically to the tumor site or to the cancer cells. So this actually so far didn't materialize into a clinical outcome properly. And that particular nanoparticle that was aimed at treating prostate cancer equipped with paclitaxel or docetaxel actually failed clinical trials in phase 2 for lack of efficacy. Instead, we started to have, also as I was doing my PhD, we started to have this type of applications where there is myth and over expectation from nanomedicine, and there are the realities. And they talk about problems, too many applications, not enough clinical outcome. a lot of research and a lot of writing came into the idea where the problems lies. Actually, the problem is everywhere, from the basic science that need to be understood behind the form of a nanoparticle and how it moves, how it interacts with its surrounding, to the preclinical, medical preclinical science, to clinical interpretation, and to over expectations by the market, not enough financing. Many problems led to this stagnation. Too many research published preclinically and not enough clinical translation. However, the first important nanomedicine that kind of tried to achieve the vision was Doxil, approved in 1995. It was a conventional liposome where it has a lipid bilayer. The lipid bilayer actually makes a cell-like structure. Inside it, we put doxorubicin, and it makes use of the specific tumor microenvironment, specific tumor architecture that is different from normal cells, where there are bigger gaps in the vasculature and not homogenized cell structure. So they favorably leak into the vasculature of the tumor, and that was the reason behind its success. Those particles were still hidden from the immune system because of the polyethylene glycol, but there was absolutely no form of active targeting, no targeting moieties that will lead them to the target site. The use of lipids was kind of a primitive use of lipids. They just needed to form the structure and to delay the release of the drug. They just basically needed to help to hold the particle together. After Doxil, several breakthroughs happened. The discovery of ionizable lipids, positively charged lipids. And then the discovery of ways to industrially manufacture these nanoparticles, namely using microfluidic systems. In 2018, a little before the pandemic, we have this drug, Onpatro. Onpatro is a lipid nanoparticle that contained RNA, so somewhat very similar to the COVID-19 vaccines. It was approved for the treatment of polyneuropathy in the hereditary transthyretin-mediated amyloidosis. This drug costs a lot of money per treatment, and this is a very rare disease. Total population is estimated to be 50,000 worldwide. Meanwhile, Moderna and BioNTech that developed the vaccines, they both did not have any commercial product yet, and no profit at all. So a little bit of comparison. Onpatro, although it was not a treatment for cancer, neither the COVID vaccines, they all share a same structure. However, Onpatro was an intravenous, and it was injection rather than intramuscular, and this is very important, and it was targeted at the liver. It used as siRNA, small interfering RNA. It's aimed at silencing RNA in the cytoplasm of the cell rather than mRNA that is aimed at manufacturing the or synthesizing the spike protein. Its primary target was the hepatocytes, so it had to travel through the blood and go to the hepatocytes, rather than the COVID vaccine that primary target was the antigen-presenting cells that actually home by themselves. They have their targeting technique and they come to the muscle where the injection happened, and the action was very topical in a way. While here we have the hepatocytes and we have a systemic application. So this is very important. This is a milestone in cancer in nanomedicine that holds some promise for cancer. Though that's not a perfect milestone, we did not have that targeting that goes to a specific site in the body because of a ligand that we equip the particle with. Actually, nanoparticles tend to accumulate in the liver. That's one thing. And they had this term, exogenous targeting, as they realized the reason behind the accumulation in the liver is that the lipoproteins themselves, the ionizable lipids themselves in the lipid nanoparticle actually have an affinity to the apolipoprotein E that exists in the hepatocytes. So to sum up, before the pandemic, an extensive theory and publication was there. And the only example of a targeted medicine that is not completely targeted in the way that we want and we can control was Onpattro. However, there was always difficulties in production at scale. Up to Onpattro also, that why they can justify charging such a huge amount of money for the treatment. And only Onpattro, there's a very small clinical outcome and safety data for the concept of lipid nanoparticles. What changed in the pandemic is that we had a big surplus of production capacity. So in January 22, they already distributed 3 billion doses of both Moderna and Pfizer vaccines. We had extensive safety data coming from clinical trials, coming from pharmacovigilance after the application. We know basically that the formulation itself is safe. We have also the expertise that being built, the supply chains for the materials. And now the Moderna portfolio of mRNA drugs has actually grown exponentially. Still nothing in the market besides the COVID-19 vaccines, but there is a list. I'm not sure if you can see much here. There is a list of cancer medications using lipid nanoparticles encapsulating mRNA vaccines. Some of them are reaching phase 3 clinical trials. But we can always hope that this is not just a promise to go to Mars by Elon Musk. And actually, the gentleman from Accurine from the first failed clinical trial is still there. He is the one who became a billionaire from the COVID vaccines, Dr. Robert Langer from MIT. And he is the one, he's a big contributor to Moderna. So I want to talk about how I envisage the future direction. First, the ideal nanoparticles have not changed much before and after the pandemic. A bit of changes. Surface functionalization is still there. Polyethylene glycol pegylation is very important and will still be important. There is no reason in my opinion, there is no reason to think about an alternative. There is a concept proposed by lipid nanoparticles that is the nanoparticle stiffness. A lipid nanoparticle is soft, while polymeric nanoparticle, a polymer, is hard. So this is a kind of there is the measurement, and there is a lot of literature now talking about the stiffness of nanoparticles. The softer nanoparticles, the easier for them to leak through the fenestrations of blood vessels and accumulate in a tumor site. Size has been emphasized in a way from COVID vaccines that is below 100 nanometers is better for drug targeting. And another aspect I want to talk about is pharmacokinetics. Actually, nanoparticles are about pharmacokinetics, not much about pharmacodynamics. We need the drug to be concentrated in the organ of interest, in the site of interest, and to avoid other sites. So far, if you do an animal study, albeit there are ethical challenges, in the animal study, at the end, you can measure the concentration of the drug in the blood, and you can also study the organ distribution. You can take out the organs and analyze. And this is what we are doing currently in our lab. However, in humans, this is unethical. You cannot take parts of humans to analyze. Or in animals, you can sacrifice and take. I want to talk about the physiologically based pharmacokinetic modeling, which is been there for a long time. And with the advancement of in silico research and AI is going to get more and more important. Basically, in PBPK modeling, it helps us predict the tissue concentrations of a named entity, drug, or nanoparticle. Originally, it was designed for small molecule drugs. From knowing we know that from knowing basically the blood concentration of the drug. So we have parameters like the blood flow to every organ, which is different, and the tissue partition coefficient, which can be taken from literature or can be taken from animal studies. And then we have a membrane permeability coefficient inside the tissue of interest. So even the tissue itself is divided into two compartments, extracellular and intracellular. If we want to, this is a model, a generic model proposed for or used extensively for small molecules. If we want to change it into nanoparticles, we have to incorporate two layers. So one layer is the distribution of the nanoparticles itself, and the other layer is the distribution of the drug, the free drug released from the nanoparticles. And this is also specific to every organ, depending on a few factors, namely, most importantly, pH, actually. Me mentioning the physiologically based pharmacokinetics is because of their importance, and according to our literature review in our specific type of nanoparticles, no one actually performed physiologically based pharmacokinetics before. It is not well utilized, and generally I find the expertise lacking in Malaysia, whether it is conventional or for nanomedicine. So this is something important to take into account, to bring the expertise, to invest in this expertise. And one thing that it helps with, in my opinion, is developing the legislative framework for nanosimilars. Much like normal bioequivalent studies needed for conventional drugs, nanomedicine has specific requirements for bioequivalence. It's not exactly the same. We cannot predict easily the concentration in the target organ compared to just by knowing the concentration in the blood. So one way easy for us to access nanomedicine is by owning the technology locally here in Malaysia is by making making generics for currently available drugs. And that is can be a lot easier just by using by developing the proper framework and acquiring the technology. In my opinion, physiologically based pharmacokinetics are important to achieve this concept of similarity between two nanomedicines. So some take-home notes: mRNA vaccine development presented a great opportunity for cancer nanomedicine research. Lipid nanoparticles are front runners in this technology. It is important to look at industrial scalability as you are conducting your research. Pegylation is still a gold standard. We have to understand that in silico tools are important, especially PBPK modeling. And I think developing the regulatory framework is important for the future in Malaysia. And that's all, and thank you very much.
Thank you so much, Dr. Hassan. That was a really great sharing, and thanks for enlightening us on the journey and application of nanoparticle in the current settings. So let's see if we have any questions from our attendees today. Maybe we can start off with a few basic questions. You mentioned about the different nanoparticles, and do you mind, telling us or what is the differences, or how do you compare a polymetric nanoparticles with the lipid nanoparticle?
Now the trend is lipid nanoparticles. Actually, it's a great opportunity to be able to work with lipid nanoparticles because of the pandemic and because of the proof that we have from the COVID-19 vaccines. There are still advantages and disadvantages to both sides. I personally work on polymeric nanoparticles as they are easier to manufacture. We can control them more in terms of the drug release. But the stiffness that I mentioned here, the nanoparticle stiffness is a measure that goes to the side of the lipid nanoparticles. Basically, however, although now the trend is lipid nanoparticles, I am still in favor of working on polymeric nanoparticles as they are much better in terms of encapsulating hydrophobic drugs. That is a basic need in drug discovery in a way. That is still that everyone gets an isolates a new compound or works on a plant extract, they will realize that okay, the new drugs that we are discovering, they are all hydrophobic and they need to be delivered in a suitable way in the body.
Okay, thank you so much, Dr. Hassan. So maybe another question. We've been hearing a lot about CRISPR. So what do you think about delivering CRISPR using nanoparticles?
My thoughts actually that the claims about CRISPR delivery using nanoparticles are pretty much exaggerated. Usually they are getting us into the same limbo of too many preclinical research that is not going to be translated properly because CRISPR basically, I mean, there is different variations of the. I'm not the expert in CRISPR itself, but what I would say, if you want to deliver a protein, it's much more difficult to deliver compared to delivering a piece of RNA, and then you need to develop deliver that protein to the nucleus in the human body. So it needs to home to the target site, to the tumor, and it needs to be delivered in the tumor to the nucleus. Delivering to the nucleus is a much more difficult ordeal compared to delivering to the cytoplasm. That's why all the studies and clinical trials actually that are now, majority of them are ex vivo, basically on T cells. They take them out and then they deliver. I think some of these clinical trials use lipid nanoparticles, if I'm not mistaken, and only ex vivo, not inside the human body.
I see. So we are afar from delivering, CRISPR to using lipid nanoparticles. There are several clinical trials, isn't it?
I think all of them are ex vivo.
Okay.
In vivo, probably, if there are some in vivo delivery using viral vector,
I see.
not using nanoparticles.
So, I guess that's it. There's no other question from the audience. So, but before we end our session, we can have another 3 minutes if anyone would like to ask anything. And also a quick reminder to all our audience to click on the link to get your CPD points. Oh, we do have a hands up. All right, it's from Prof. Sarah. And also from Prof. Sagrunan. Okay. So a question, we have a monoclonal antibody and a small molecules in autoimmune diseases. Will they be advantages here in terms of cytokines and cell targeting?
You mean advantages encapsulating them in nanoparticles?
Yeah.
If I understand the question. Well, monoclonal antibodies, if there is off-target effects, as I assume, yes, it should be an advantageous to encapsulate them in lipid nanoparticle. Monoclonal antibodies are an example of a molecule that actually favors lipid nanoparticles because it can be encapsulated in the core that is hydrophilic. So maybe we can, I think that yes, we can, and it will reduce off-target effects. If it did not manage to increase the effect on target, at least it can reduce off-target effects in theory.
Thank you, Dr. Hassan. So we have another 1 more minute and last question from Prof. Sarah. Do you think that Malaysia can make use of this technique to compete and be a world player in cancer treatment? Where are we now and how do we get there, basically?
I do think, yes, we can, if not mastering the whole chain, all of it, we can work on some parts of it. Like in Malaysia, there are people who work on microfluidics, there are people who work on lipid nanoparticles. There is already a lot of research. My understanding is that we have to focus on it. And if it's not cancer, other applications of lipid nanoparticles. And if we cannot master all the techniques, at least we can try to own some of them and make use of it. The development of microfluidic techniques to manufacture nanoparticles, the development of the lipids, the positively charged lipids, that they are still trade secrets for the companies. Those are important, I think, sides that we can take parts of this work and focus on it and develop it, at least.
All right. Thank you so much, Dr. Hassan. That's a brilliant sharing. I hope that all this insight would open all of us, all these attendees here, to be willing or have an idea to work with nanoparticles and reach out to Dr. Hassan if you have any other ideas or discussion on this matter. So with this, we will end our breakfast session this morning. And thank you to all our attendees for tuning in until the end. Thanks, everyone. Have a great day ahead.
Thank you. Thank you, Dr.
Thank you, Dr. Hassan.